Okay, hello everyone. I hope you all had a good second spring break, and we're into the home stretch. Hoping this last unit will be our latest one so far, but we'll see how that goes. Last exam looked good by and large, so we should be ready to get rolling. All right, so today we're going to start out thinking about the GI system. So this unit is going to be finishing out our human body system. So we're going to be talking about the gastrointestinal tract and digestion. And then we'll be talking about the reproductive system before we finish the course. Okay, so today we're going to start out by thinking about the anatomy of the gastrointestinal system, which hopefully will be somewhat familiar to you, as I think you've already seen it in lab. So we'll go into a few more details than you can see physically in the donor body lab, but some of these terms should be relatively familiar. So we're going to start out by going through tissue layers and major organs of the GI system. We're also going to end up going through some accessory glands that we're going to see as we start to talk about the digestive process. Okay. So overall, process-wise, what we're thinking about, when you eat food, it is too big to just pull into a cell, right? You can't pull a whole cheeseburger into a cell, not possible. So consumed nutrients are too big, too large to get directly into your bloodstream, right? Not possible. So the process of digestion is the process of breaking down your food and even breaking up the molecules within your food until they're small enough that they can actually cross these barriers, cross these cell layers, and get from your digestive system, from your GI tract, into your bloodstream, where they can then circulate around and get to other cells in your body. Okay. So as we go through, we're going to be talking about that digestive process, that breakdown of the food, that breakdown of those molecules. We'll think about how those different types of molecules get absorbed across the walls of the GI tract and then into the bloodstream. And we'll also talk a little bit about how the structure of the GI tract allows food and the things you consume to move along. So that's the motility. Motility is movement. And we also have secretion. Secretion is going to be really referring to the fact that we're going to be pumping a bunch of enzymes into your GI tract to help with the chemical breakdown of these molecules that you're going to need in order to digest and ultimately absorb the nutrients in your food. Yeah. So we see here that we have two systems that we're trying to link together in this sort of peach color. We have organs of our GI system. So we see our GI tract here from food and water at the top to the rectum and anus here at the bottom, getting rid of unabsorbed material. And we're noting that what we're trying to do is absorb, so move from the GI tract into the bloodstream. So moving into the bloodstream here is our absorption process. Okay. And in this process, we're also going to be secreting, so moving some things from the bloodstream into the GI tract. Okay, so these are processes we'll see as we go through. Okay. So we're going to begin by thinking about just the divisions of the gastrointestinal system. So we're going to be thinking deeply about two different parts, sort of. So we're going to have the gastrointestinal or GI tract. Think like a track, like a pathway, right? So the GI tract are the hollow organs that your food is passing through, essentially. So about 15 feet long, begins with your mouth. So you're going through your mouth, through the pharynx, down through the esophagus, into the stomach. Food churns around. Then we go through your small intestine, your colon, and finally out through the rectum and the anus. Okay. So these parts I think you've seen in lab. And we do have accessory glands along the way. So these are going to be important for secreting fluids and especially enzymes that help the breakdown of food into the GI tract. So we'll go through those as they're relevant, but things like the liver, the gallbladder, the pancreas are going to be important as we move through. Okay, so those are also part of this whole system, even though they're not hollow tubes They're not part of the tract itself. So when we're thinking about that tract, that hollow tube, we could look at the walls. Right. So when we're looking at the wall of the GI tract, we'd see that it has several layers. Okay. So lumen, this word lumen, we've seen before, we may or may not have concentrated on, but lumen, remember, just means the hollow center of something. That's what a lumen is. Okay. So noting the lumen here is in the center, and we've cut through in two different sort of directions this wall of the GI tract. Okay. So your food would be traveling through the lumen, and directly lining that, directly touching, you're going to have the mucosa, the lining of the lumen. So you'll have mucosa lining this space. Okay. So we'll see that the mucosa actually has 3 parts other than just that cell layer. Okay, and we'll talk about those in a second. We're talking big layers first. So that's our mucosa. Next, we have our submucosa. This is really a layer of connective tissue. So stuff connecting, holding stuff together. That's our submucosa. So sub, because like if we were getting from the lumen to the, which is the hollow part, to the submucosa, we'd have to dive through the mucosa. Right, so we're kind of going from the inside out. Next, we have the muscularis externa. Okay. So muscularis, because we're going to have some muscle there. Okay. Specifically, we're going to have smooth muscle here in this region. Okay. That's going to be important when we think about sort of squeezing food along, right? It's important that we have muscle in the wall of our GI tract. And then finally, we have the serosa. Okay. The serosa is another layer of connective tissue. And it's going to be kind of slimy if you think about this word like serous fluid. I don't know if anybody's mentioned that to you. I'm not sure I have. But it means it's kind of like covered with a slimy coating so that your intestines can kind of move around, squish against each other as food moves through. Right. So beginning with that mucosa, said there were some layers within it. So we're thinking about this lining area on the inside of the tract. So we're separating the lumen, the GI lumen, where your food travels, from the internal environment, by which we mean like the environment of your body. So like your blood, your interstitial fluid, all that good stuff. Okay. So we're going to have an epithelial cell lining here. So here, okay. This is going to be made up of enterocytes. So entero, think like enter, like inside, sites for cells. This is going to have absorptive cells, exocrine cells, and endocrine cells within it. So absorptive cells are going to help you absorb nutrients. Exocrine cells are going to be secreting like local stuff like enzymes and things kind of locally. And endocrine cells, we've talked about our endocrine system before. That's when we have like hormones going into the bloodstream, so kind of like long distance signals. So all of that is going on in our GI system. Okay. So our epithelial lining is there. Okay. So that's this kind of mucous membrane. Okay. Or here. Then we have a connective tissue layer. So that's this yellow part here. This connective tissue, yellow in diagrams usually means fat. So you might expect some sort of fat in this lining. Okay, so that's our connective tissue. And then we have a very thin layer of smooth muscle within the mucosa itself. Okay, so that's what these little stripes are here. That's showing us the muscularis mucosae. Okay, so just noting two things we're differentiating already, right? So within the mucosa, we have a layer of smooth muscle called the muscularis mucosae. And one of our larger layers was a larger muscle called the muscularis externa. So we kind of have 2 layers of muscle going on here. So here we see that, I guess I got ahead of myself. So we're still just tracking through that mucosa. So we looked at the mucous membrane. Now we're looking at that yellow part, the lamina propria. So that's the connective tissue. And it has running through it as well, some small blood vessels and lymphatic vessels. Okay, so you can see lymph, especially right here. Noted some lymph nodules here. There'll be nerves in here as well. Our lymphoid tissue is going to be separated into two kind of groups. So we have things we call lymph nodules, and there are also things we call Peyer's patches here, both to do with that lymphatic system. And then we had our muscularis mucosae, that layer of smooth muscle within the mucosa itself. So it's thin, as you can see on the diagram. Its fibers are both longitudinal and circular. And the job of this layer of muscle is to kind of mix around the contents of the GI tract. So when we think about moving food through your tract, you're going to have to both mix everything together. So like enzymes touch all the different parts of the contents of your GI tract. So that's what this is doing. And then you also have to push things forward. Okay. So when we talk about motility of the GI tract, movement of the GI tract, we'll be talking about those two separate but linked functions. They're both going to be necessary. Okay. So stepping out, that purple layer is the submucosa. So this layer of connective tissue is pretty thick. It's distensible and elastic, meaning it can stretch, it can distend meaning or like changing the shape, kind of stretching, bulging something out. And it's elastic, so it can bounce back. Here we're going to have larger blood vessels and lymphatic vessels kind of embedded. You can see those in the image as well. And here we have actually a part of kind of the nervous system. So the enteric nervous system is part of the nervous system that's kind of embedded in organ walls. So we have a plexus related to that here in the submucosa. It's called the submucosal plexus, or also Meissner's plexus. And so you can see some of those nerves here. Stepping out further, looking at the muscularis externa. So this is a thicker layer of smooth muscle. Also has longitudinal fibers as well as circular fibers. So these layers, instead of just mixing stuff around, we'll get to why this helps to push food forward, but it's going to have an inner circular layer that changes the diameter of our tube, right? So it changes its like diameter, its radius, so making things narrower and wider. And the outer longitudinal layer is going to change the length of the tube. So it's actually going to kind of stretch longer and contract shorter. So those are two sort of types of movement we'll be able to see in response to the muscularis externa moving. Here we have another plexus called Auerbach's plexus, or the myenteric plexus. So this is working in conjunction. It's working with that previous plexus in the submucosa. So we had Meissner's plexus, or the submucosal plexus here. And now we're stepping out and we have Auerbach's plexus, or the myenteric plexus here in the muscularis externa. So these work together. So this is the second part of our enteric nervous system, which is part of the nervous system that's like dedicated to the walls of our organs. So now we are moving out to our final layer. So the outer kind of covering is the serosa. Okay, so it does have layers within it as well. So it has a connective tissue layer inside. And then the outermost part is a layer of epithelial tissue, specifically called mesothelium. And that's going to be continuous with the mesenteries that you saw in lab. So those long, sort of slimy, potentially fatty sheets that are continuous with the peritoneum. So all of this together is allowing the contents of your abdomen to slip slide against itself, essentially, as the size of different organs, different parts of the GI tract change. Or as you breathe, as we've covered. Okay. So now we're going to move from those basics of the walls, and we're going to start thinking about the tube straight through it. So when we're thinking about the gastrointestinal tract, the GI tract, we're talking about the tube, the path with which your food is going to be following. So you're going to start in the mouth. And digestion actually starts there. So mastication is the formal word for chewing. the scientific term. So biting things with your teeth, grinding it around, that is actually sort of manual digestion, right? You're physically breaking up the food. You're starting the digestive process. And your saliva actually helps with that. So you are secreting saliva into the mouth. So you have salivary glands that you've seen, right? They have little tubes. They secrete fluid into your mouth. That saliva both helps lubricate so that you can move a ball of food around your mouth and then also down your throat. And it also includes our first enzyme that we're going to pay attention to. So the saliva includes an enzyme called salivary amylase. So hot tip remembering enzymes, right? Anytime we see that ACE ending, we're talking about an enzyme, talking about, in this case, something that's going to be breaking apart our molecules in digestion. So salivary amylase is in the saliva. And what it does is it digests primarily starch as well as glycogen. So it starts some of our carbohydrate digestion. And later in this unit, we'll be walking through in order, how do we digest carbs? How do we digest proteins? How do we digest fats? We're going to mention these things as we go through the locations where we'd find them, and then tap back at them when we talk about those from a molecule by molecule basis. Okay. So after you chew up your ball of food, you start to swallow it. That ball of food, which literally scientifically called a bolus, would start to slide down your throat into your pharynx. So we know we had the nasopharynx. Hopefully you don't have food there. Now we're in the oropharynx, right? We would pass into the laryngopharynx and then into the esophagus. So the pharynx is a common passageway for air and food until we have our two tubes split, GI tract remaining posterior, and our airways anterior going through the larynx. So we're passing from the pharynx into the esophagus. So the esophagus, going to look pink, collapsible, muscly when you saw it in a lab. It does have sphincters that control what enters and exits, how fast or how slow. So your upper esophageal sphincter, that's this constriction point, so this narrowing. It's made up of skeletal muscle, and it's a separation between your pharynx and your esophagus. So skeletal muscle, meaning it's under voluntary control, so you can open and close it, right, to allow food to pass or not. We have a muscular tube then running all the way down to our stomach. And it's going to be made up of different parts, or sorry, different types of muscle, depending how far down you are. So the upper third of the esophagus, the upper third of this tube, is going to be made up of skeletal muscle, so muscle you can consciously control. Then the lower 2/3 are going to be made up of smooth muscle, which is going to be under involuntary control. Right? Those are two types of muscle we've distinguished in this class: skeletal muscle, smooth muscle, and our third type was cardiac muscle. Right? So the skeletal muscle is the voluntary one, the other two involuntary. Okay. So we're passing through the esophagus. We have a region of skeletal muscle under our control, then we're out of our control in the smooth muscle until we get to the lower esophageal sphincter. So like the lower parts of the esophagus, the lower esophageal sphincter is going to be made up of smooth muscle, not under your conscious control, located between the esophagus and the stomach. So we're entering into the stomach as we pass this sphincter. When we're in the stomach, that ball of food is going to get stored and it's going to undergo more digestion. Okay. So the stomach changes really drastically in its size. Its empty volume is about 50 milliliters, full volume, maybe like 1,000 milliliters, so a liter. It has a lot of wrinkles, essentially, these rugae, that allow it to expand significantly and contract into the smaller volume. So rugae, think wrinkles is what those are. So that allows it to expand kind of like an accordion and contract back down. Now, while your food is in there in the stomach, it's being stored, but your stomach is also secreting things to help digest that food. And what we're going to be doing in the stomach is moving from having a ball of food, a bolus, into having a mixture called chyme. So chyme is going to be like a smoothie of food and gastric juices. And that's what we're eventually going to be passing into our small intestine. Okay. So our chyme is what happens when we have food in the stomach mixing together with gastric juices that are coming out of the walls of the stomach. So this is zooming in, looking at the wall of the stomach, looking at gastric pits, so kind of like holes in the wall that lead us down into necks and then glands, essentially at the bottom here, where we are going to be secreting enzymes and acid here. So this is happening in the stomach. And then we are going to release our food slowly into the intestine as we have a sphincter at the end of the stomach, our pyloric sphincter here. Now let's take a look at those gastric pits where we are creating our enzymes and our acids. Okay, so we've zoomed in on the stomach lining. Okay, so we took, say, a section right here, zooming in. So here's our mucosa, right? And the pit is diving down into the wall of the stomach. So they're leading into what we call gastric glands. Okay, so we can see these are labeled here as gastric glands, kind of down in the bottom of these pits. Okay. So we are going to have a couple of different cell types here, and we are going to be producing things. So we're going to have chief cells. Those are these yellow ones, which are going to secrete something called pepsinogen. We're going to have these purple cells called parietal cells, which are mainly going to secrete acid. So the pepsinogen, some parts of this word might be familiar. So we've seen peptides, right? So maybe this pepsin part might indicate to you this is going to have something to do with proteins. It is. It's going to help us digest proteins ultimately. We've also seen this ending before, this inogen or ogen kind of ending. I told us that this is something kind of inactive, right? So we're going to have a flow of enzymes as we move forward, kind of activating this eventually. So this is a precursor molecule for something that's going to help us digest proteins as we move forward. So that's the pepsinogen. And then we also have our acid, which is really just hydrogen ions. So we talked about pH at the end of our last unit. We talked about how acidity measured by pH is kind of excess hydrogen ions. We talked a lot about carbon dioxide because that was our important respiratory acid. But now that we're in the stomach, we're thinking about hydrogen ions. So lots of hydrogen ions are our acids coming out of the parietal cells. Okay, so we're releasing a bunch of hydrogen ions here. And this is why your stomach has such an acidic environment. And this does help with the breakdown of food as well. But you may have felt this, right? If you've ever had a really nasty stomach bug, right? And you keep throwing up even when there's nothing in there, feels very acidic, burns your mouth, right? It's because it is, right? There is strong stomach acid in your stomach coming from those parietal cells in the gastric pits. Another thing that those parietal cells are secreting, which is not on this figure, is something called intrinsic factor. So this is going to be necessary for absorption of vitamin B12, which is an important vitamin for your body. Also not pictured are gastrin molecules. Gastrin is a hormone that gets secreted from G cells, which are also down here in your gastric glands. And those ultimately go into your bloodstream because we're going to need to do things like send signals to your brain about how full is your stomach, right? And to send a long distance signal, we need to put something into bloodstream. We need a hormone. Finally, we have our mucus cells up at the top. So our neck cells are secreting mucus. just going to help kind of protect our stomach from that high acidity. So we're secreting a bunch of acid from those parietal cells to help digest our food, but we don't want to literally digest our own stomach from the inside out. So mucus is important in the stomach, partially for that reason. Speaking of that acidity, so the pH of our stomach is 2, very low, right? So when we were talking about normal blood pH, our floor for that, our lowest normal pH was like 7.35, right? The acidity of the stomach is much, much, much more acidic. And this is necessary for digestion, partially because this is helping us to activate that precursor molecule for our protein digestion, that pepsinogen. So literally that acidity is part of what activates this, okay? So that's one thing that acidity is doing. It's also helping denature proteins. So when we talked about proteins way back when, we talked about their 4 levels of structure, right? How we link together amino acids in polypeptide bonds. Then we started to fold into our helices and our sheets, right? The alpha helices, the beta pleated sheets. Then we crumple up into a complicated kind of 3D structure based on their variable side chains, their R groups, right? And then we also had subunits coming together. So maybe one polypeptide crumpled into this ball, another one like this. And we had subunit interactions, like when we looked at hemoglobin, for example, right? It had 4 subunits within it. Okay. So when we say that something denatures proteins, what we're saying is that we've made a change in the environment that causes those proteins to kind of unfold and start to break, right? So when we make the pH of the stomach a pH of 2, that really changes how, for example, the different R groups interact with each other. It disrupts those associations between parts of our proteins and kind of helps us start to unfold them. So that's partially a reaction to the pH, to all these little hydrogen ions floating around. Also kills bacteria, taking dunked in acid, who knew? Right, so that's kind of an immune function really here. And we mentioned that our neck cells were secreting mucus. So we technically call that barrier that they're building up the gastric mucosal barrier. So it's mucus, which is the slimy fluid part. And there's actually also bicarbonate in it. So bicarbonate is a buffer for acidity. Right? We talked about that when we were talking about pH balance in the blood and the kidneys. So right here, when we put bicarbonate in our mucus layer, that means when those hydrogen ions come into contact with it, it can kind of help buffer it out there. Okay. So those are coming from our neck cells, as well as some cells called goblet cells. So now we're going to move from the stomach into the small intestine. So we're passing, we had our esophagus, our lower esophageal sphincter. We had our stomach. We mixed everything together into food plus gastric juices. So now we have chyme. We're going to pass from the pyloric sphincter into the duodenum, which is our first part of the small intestine that we'll continue to walk through. Now, your small intestine is really adapted for absorption, and it's really efficient at this. Absorbing all those nutrients from your food should be happening in like the first 20% of the intestine length. You have a lot of extra, which is pretty good, right? We do want to catch all our nutrients. Your intestines are structured in such a way that provides a really large surface for absorption. Now, we need a large surface area because it's great that we can break down our molecules, right? Make them small enough to get into the bloodstream, but then they actually do have to contact a wall to physically cross it and get into the bloodstream, right? If they just stay in the middle of our tube, that's not going to help us, okay? So we're going to have a large surface area so that all these molecules that we're digesting, breaking apart, bump into the walls, go into your bloodstream. So beginning with the small intestine, this is a coiled hollow tube, 8 to 10 feet long. located between your stomach and your large intestine. Okay, so stomach here, large intestine beginning like here. So all of this stuff, right? I'm obviously not tracing the actual folds here, but that's all small intestine. Okay? So this is our primary site for digestion and absorption. So in this first 20% of your intestine length, we have three parts. So we have the duodenum, the jejunum, and the ileum. Those are in order. Now, before we trace through our food going through, we're going to look at the wall of the small intestine specifically. So I mentioned that we are increasing surface area for absorption. So one of the features that lets us do that is our small intestine has these structures called villi on it. So you can see these projections up, these extra little kind of nubbin things. This increases surface area compared to, right, if we just had a flat layer of cells. Right? So they're increasing the surface area of the epithelium. They also contain blood vessels and lacteal for the absorption of nutrients. So you can see the blood vessels going in. So that's going to be our arterial going up here into a capillary bed. On the other side, we have a venule going back to our vein. So we're going into the circulation that way. And a lacteal is like a projection of the lymph. So you can see in green, green is the color your book likes for lymph, by the way. So we have a lymph duct going through, and it has an analogous structure called the lacteal that's also projecting out to here. So you got all three things together. You have an arterial, a venule, and a lacteal in here. All of those are going to be involved with absorption, depending on what type of molecule we're trying to absorb. We look at the cells themselves. You see a similar structure on the apical membrane. So we saw this also in some of the cells in our kidney. On the apical membrane, we have microvilli. So we see those there. So the villi, the big ones, would be made up of multiple cells, right? Like every one of these boxes would be a cell along it. And then each of the individual cells within themselves also has projections, but they're little, they're on a cell. That's why they're the microvilli versus the villi. So this is forming what we call a brush border because it looks like the surface of a brush. So this is all just about like duplicating surface area over and over again so that we have lots of room for absorption. Another thing we have here in the wall, you can see between these projections, these villi, we have what looks like pits again. So these are our crypts of Lieberkühn. Okay, so crypts, like cavities, like, yeah, they're crypts. So there are epithelial cells lining these crypts as well, okay? And they are going to be secreting a fluid that's really rich in bicarbonate, okay? This is mostly going to be happening in the proximal small intestine. If you step back and think about what's going on, this may make a bit more sense, or at least be a little easier to remember. All right, so in the stomach, we mix together this strong acid with our food to produce this chyme. Right, so that means that the chyme, which is what we're going to end up having to squeeze into our small intestine, is also going to be highly acidic, right? We had a pH of 2 in the stomach, and then we're just squeezing the contents of the stomach into the small intestine. So we're squeezing a fluid that's highly acidic into the small intestine. So the crypts of Lieberkühn secreting this bicarbonate-rich fluid are essentially protecting your small intestine, helping us buffer out that acidity, again, protecting the tissue of the small intestine itself as you move forward, okay? So this is protecting tissue. So it's getting secreted in the proximal intestine. By the time you get to the distal small intestine, you digested all the food, you've mixed around the products, you have secreted this bicarbonate-rich fluid in the beginning of the small intestine. We like keeping bicarbonate around, right? Because we like to reuse it. It's a great buffer in our bloodstream, as well as now we can see in our GI tract. So in the distal small intestine, you're going to reabsorb essentially that bicarbonate. So it gets absorbed in the distal parts, so the farther along parts of the small intestine. Right. So we had our bolus of food, we chewed in our mouth, swallowed through our esophagus, passed through our lower esophageal sphincter into the stomach, mixed it up with our gastric juices. So now we're calling it chyme, squeezed through the pyloric sphincter into the duodenum. Okay. And now we're also going to have other secretions into the duodenum, in addition to the fact that we know that the tissue in the walls of the small intestine in general has these crypts of Leber-Kuhn secreting those fluids with our bicarbonate ions in them. So we also have juice from the pancreas coming into the duodenum. So we're going to have a duct opening into the duodenum from the pancreas, just kind of nestled behind the stomach. Okay. This pancreatic juice is going to include more bicarbonate. And this is helping us neutralize essentially that acidity of the chyme coming from the stomach. The pancreatic juice is also going to include a lot of digestive enzymes, helping us break up the molecules in our food, helping us, break it apart into small parts that we are then able to absorb in this big surface area lining our intestines. The other thing that's going to get secreted into the duodenum, so squeezed out from somewhere else, is bile. So bile is coming originally from the liver, secreted by the liver, can get stored in the gallbladder. So you saw that's often a really intense green, right? So those bile salts are really important for fat digestion. So when you want to digest fats, that's when you're going to use the bile, squeeze it into the duodenum, mix it in with that chyme coming from the stomach that will include fats, and those bile salts are going to help with the digestion of those fats. So that's happening up here at the duodenum, so the first part of the small intestine. So we mentioned your liver once or twice before. One of the things that we said was important about it was that it was creating our clotting factors for helping us create blood clots. But the liver is really important in absorption as well for a lot of reasons, actually. So one thing that's true about the liver is that when we're looking at the blood flow to the intestines and the GI tract, right? So we're absorbing our nutrients into capillaries that are ultimately going into veins, right? So to get to your body, anything that you've absorbed through the walls of your intestines has to travel to veins, back to the heart before it gets pumped around, okay? But we've seen that the veins in our abdomen have this weird kind of structure, right? They don't directly immediately go back to the vena cava, right? A lot of them are going back to the hepatic portal vein instead, and they have to travel through the liver first before they get to the vena cava by the hepatic vein, so before they get back to your heart. So this means that the things that you're absorbing from your GI tract into your bloodstream are traveling through the liver first, essentially, before they actually get to your systemic circulation. So this means that the liver has the opportunity to detoxify substances. So this is like where we process alcohol, right? I don't like when people just mention toxins randomly without specifying what they're talking about. So I'm giving you an example, right? So it can detoxify things like neutralize alcohol, right? It can process certain nutrients, tag little molecules, things like that, as well as being the source of the bile and these bile salts that we need for our fat absorption. Okay, so it's doing a bunch of stuff here. So details on that hepatic portal system, right? So the vasculature that's delivering absorbed nutrients to the liver, it's called the hepatic portal system. These nutrients have to go through before they enter general circulation. The nutrients originally are getting absorbed from the small intestine into the mesenteric veins. They're traveling from those mesenteric veins to the liver. They go through the hepatic portal vein to get there. And then they travel from the liver out through the hepatic vein to the vena cava and then back to your heart before they then get to travel around with the rest of your blood. Your liver is also supplied by an artery. So we have a hepatic artery supplying the liver with fresh blood and oxygen. So the liver isn't like getting its blood from the hepatic portal vein. It has its own blood flow from the aorta. So after we go through the small intestine, so we went from our stomach to our duodenum, jejunum and ileum. Now we're in the large intestine. So we're going to have our cecum, our colon, and our rectum. Okay. So the beginning point is going to be our ileocecal sphincter. So between the terminal portion, so the end of the small intestine, the ileum, and our cecum, we have another sphincter. We're going to note that our large intestine has new bands of longitudinal smooth muscle called tinea coli. So you can recognize large intestine from small intestine if you notice those longitudinal bands, the tinea coli. And the colon as a whole is just a big hollow tube. It's going from your small intestine to your rectum. Has its sections. We were beginning at the cecum, then we go through the ascending colon, the transverse colon, the descending colon, then the S-shaped sigmoid colon, ultimately into the rectum, and then out the anus. And we have done the absorption of our nutrients in the first 20% of our intestines, right? So that was really happening in the small intestine. So by the time we get to the colon, we don't really have to absorb nutrients anymore. What we do want to do is concentrate waste into feces. So we want to absorb most of the water. A lot of that is going to be going on in the colon, kind of solidifying the feces, and then store the feces until defecation. You don't want it just like dribbling out all the time. We want to be able to store it. Okay, so that's going to be the job of our colon. The colon empties into the rectum, and then the feces gets excreted through the anus, which, lucky for us, has sphincters. There's an internal anal sphincter made of smooth muscle and an external anal sphincter made of skeletal muscle. So kind of paralleling what we saw with the urethra in our urinary tract. We have to relax both sphincters to open this tube, right? So to allow for defecation, pooping, excrete feces, we have to open both the internal anal sphincter and the external anal sphincter outwards. Okay. So mentioning some glands now. Okay. So mention some of these as we were going through talking about where their products are going, but let's take a look at them individually as well. So the accessory glands of our digestive system, where we're going to be secreting products, enzymes, bicarbonate, we're going to be making them, secreting them into the GI tract to help with our digestion. Okay. So we're going to have 4 types. So we have salivary glands that secrete saliva. We have the pancreas secreting pancreatic juice. We have acini. So those aren't going to be like a separate organ. Acini refers to the fact that in a bunch of glands, we have these little sort of bubble looking structures. So the acini are those. So they're clusters of secretory cells made of epithelium. Okay. And then we have the liver, which secretes the bile. Okay. So the general process for secretion is that we're going to make whatever we're secreting, primary secretion. It'll get modified as we travel through a duct, and then eventually we'll go into the lumen of the digestive tract, so into that hollow area where we have our food traveling. Okay. So when we think about saliva, so when we mentioned the mouth for chewing, right, the saliva contains mucus. We mentioned that we have salivary amylase for thinking about the saliva in your mouth. We also have something called lysozyme, okay? And we also have a bunch of bicarbonate ions in saliva. So saliva is fluid mixed with all these things kind of suspended in it, mixed together. When we think about the pancreas is going to have both exocrine and endocrine portions. So we actually talked about the pancreas before when we were talking about the endocrine functions, right? That's when we were talking about like our islets of Langerhans. We were talking about glucagon and insulin. That's the endocrine function of the pancreas. So the exocrine function of the pancreas is what we're thinking about now. Exocrine referring to the fact that we're creating these pancreatic juices that have these enzymes for digestion. Right? So those are two separate, both important functions of the pancreas. Produces hormones as part of its endocrine function, produces these enzymes and bicarbonate as part of its exocrine function. Okay. So the pancreatic juice is going to be rich in bicarbonate, lots of bicarbonate in it, which is great because we're trying to neutralize that chyme coming from the stomach, super acidic. Relative to the weight of the pancreas secretes more protein than any other tissue in the body. That's just kind of a fun trivia fact. When people think protein, they often think like muscle, right? But the protein it's excreting is really like all these enzymes it's making, essentially. So from our pancreas, we're going to have another amylase. So we're going to have pancreatic amylase helping with carbohydrate digestion. We're going to have various lipases, proteases, and nucleases helping us break apart our lipids, our proteins, and even nucleic acids. So those are all going to be mixed together in what we call pancreatic juice. Okay. So we'll stop here for now, finish out with the liver next time. Before we move into, make sure these are in the right order.